questions. He's going to give a talk on the immune system. So we all have quirky immune systems here, or we've seen that, right? But I do want to tell you a couple announcements coming up. So today is tomorrow night on Thursday. Actually, Dixie has that right in her hand. At La James from 5 to 7 p.m. We're going to have a booth there. And I was just informed on Saturday we were asked to have a booth at, it's a 5K, it's a Halloween. I'll tell you about that later, Abby.
I'm surprised.
I know. So it's a booth down at 1031. It's a fitness place, and they're doing a 5K down on State Street to the East Village and back loop, and then you can do like almost like a pump and run, like the QC Marathon does. So it takes away from your time, which will be nice. You're fine. Do you want me to throw the seat over?
No, I'll go back here.
And so that'll be on Saturday, and you can sign up the day of if you're interested in doing a 5K. It's $25. And we're just going to have a booth there just to have the awareness. And then our next kind of bigger event coming up is Dancing with the Stars, Dancing with the QC Stars. So we have about 23 celebrities in the Quad City area. And we have doctors, lawyers, radio station people, you name it. So it's going to be lots of fun at the waterfront on December 9th. The doors open at 6 p.m. And then there's VIP tickets. There will be general admission tickets. The tickets are $25 for general admission. The VIP are $45, which includes some wine and beer and heavy hors d'oeuvres, and of course the show. And then we also have tables for sponsorships for $500, a table of 8. So if you're interested in that, let me know afterwards. And so that's kind of coming up this year. We also have a cooking class, of course, on November 9th, and that's at Hy-Vee on 53rd and Devil's Glen. And then it'll be the second Monday of the month on December, I should have my notes in front of me. What is that day? The second Monday of the month on, I think it's December 7th, if I'm correct. But, well, the 9th is a Wednesday, so it's the 7th, yes. Right? Yeah, so December 7th will be the next, and that'll be a make and take, again at Hy-Vee. And then on November 23rd, instead of the last Wednesday of the month in November, because it's the day before Thanksgiving, we are going to have our healing meeting on November 23rd, which is a Monday. And we will have Mark Hippler from the Abbey, so he'll be counseling, and it'll be a small group, kind of small talk, just informative, very informative, so that'll be great. And so that'll kind of end our year, and then we can talk about next time about January and so on. So do you have anything else to add, Abby? No. So I'm going to introduce Dr. Staub. He has been my doc for over 20 years, and I was blessed to have him, diagnose me, and he's just been a wonderful man, very knowledgeable, and so we're blessed to have you here tonight.
Glad to hear that. Lisa, I want to leave a little time for questions at the end, and so the talk plus the questions should end by what time?
That's up to you.
Well, just give me a rough idea.
Do you feel 30, 40 minutes is?
Yeah, I could be done with the questions and everything by 40 minutes.
Okay.
Should we do that?
Great.
Yeah.
Sure.
So, no later than 5 to.
We won't kick you out or anything.
Okay. All right.
Do you want me to dim these a little bit?
Yes.
That work?
We had a little trouble getting the thing to expand, unfortunately, because the Macintosh and the Apple Power, or the Macintosh and the Microsoft PowerPoint don't
Does that
seem
help
to
a little
want
bit on that side?
to totally go
I think we can see that pretty good.
Should we turn this one off too?
Yeah.
Would that be too dark?
There's enough light
Maybe
coming
better,
from
I
the.
think. Our eyes will get used to it. There's some pictures that'll be hard to see with that big light showing, so this is preferred from my point of
Perfect.
view.
I
Yeah.
have little dimmers.
Turn that thing on, yeah. That's good.
Yeah. So the question might be, why talk about the immune system? It's not really a clinical kind of a talk, but the reason is because really the immune system, immunology is the heart and soul of rheumatology. The autoimmune diseases are diseases that relate to aberrant function of the immune system. So this is really the heart and soul of our discipline. And in rheumatology, when we see a patient, we try to decide at the front end, is it inflammatory, that is the immune system gone awry, or does the person have something non-inflammatory like some kind of a tendonitis or muscle strain or something like that. But the half of rheumatology that really gets the most interest and the most press really is the half that relates to the immune system. So it's good to think about the immune system. There have been a lot of really amazing things that have come about, knowledge that's been acquired over the last years since I was in medical school. I graduated in 1982, and there's been a ton of things that have come out since that time. The basic idea is it's a network of players cooperating together, and some have compared it to a football team, and a football team trying to operate together. If you were to just isolate the tight end and watch him only and see him draw off a couple defenders, and you wouldn't realize he was opening up a hole for somebody else on his team to go. So it's looking at any given little part in isolation, you kind of don't really understand. It doesn't even make sense. But the whole thing, and that's what I want to try to do, is just kind of give an overview of the whole thing from a more of a 30,000 foot sort of level. But having said that, unfortunately, it's a very complicated system, so there's a lot of details, so it's hard to totally divorce yourself from the details when you're trying to look at it in a broad sense. So I'm going to try to cover the major details that are really important. The purpose is to protect the self, that is the individual, from microbes in the environment. And in the process, the self tissues have to be left untouched, undisturbed. So it would be like in the football analogy, if you have the home team, they're on the field and they're fighting the opponent, that's great, but if the home team started tackling the home cheerleaders and the home fans, that would be bad. So this is kind of what happens in autoimmune disease. So the broad categories of microbes that really the immune system evolved to be able to handle are bacteria, viruses, and parasites. And I mention this because it's important to know the enemy in a sense, because then you can figure out, it makes more sense how the immune system is operating if you understand some basic features of the enemy. And these three categories do differ from one another. Viruses, some people would say, some researchers feel are not even really in the strict sense living items, because they are just the genetic material inside. It could be either ribonucleic acid or DNA, and then on the outside is a thin protein coat, and that's it. So they cannot live alone. They just have no cellular machinery to be able to synthesize proteins and be able to stay alive. They pretty much are in some ways of defining it not really living, apart from being able to be in a host, and then they can take over the host cell machinery and really get going and cause all kinds of trouble. Bacteria are one cell, very simple microbes, and they're called prokaryotes because they don't have a nucleus that's really defined by a membrane. So eukaryotes, that's like us and higher animals, have a nucleus that is bounded by a membrane, and within that is the chromosomes and the genetic material. And then outside of that nucleus is all the cellular machinery, and then there's a outer membrane of the cell itself. So bacteria just have basically the outer membrane, and they have no real nucleus that's easily definable with the membrane. And E. coli is an example. So we all have E. coli living within us, and it's a part of our normal flora. And sometimes there can be certain strains of E. coli that cause problems. You remember years ago there was a Jack-in-the-Box episode in the West Coast where somebody ate a hamburger that had E. coli in it, which was a strain of E. coli that was producing a toxin that was really bad for people. And so some E. coli can be bad, but plenty of it is good, and we all have certain strains of it in our intestine. So parasites are defined as organisms that live in or on a host to obtain nourishment, and they provide no benefit to the host. You could say, well, gee, isn't that true also of viruses and bacteria? Well, the fact is that in a strict sense, you could say those two items, those two categories are also parasites, but the traditional definition includes pretty much what I've listed here, the one-cell organisms, which are protozoa, and then yeasts, and also the multicellular organisms, which are fungi and worms. And it turns out that in terms of these organisms affecting us in humans, in America at least, we don't see much in the way of worms and protozoa because we have good public health. Primarily, we have good water supplies that are not contaminated. But if you go worldwide to places where they have bad, filled up water, you'll get plenty of worms and protozoa. In fact, worms are very prevalent, but we really don't have much of it. What we have mostly causing trouble is yeast and fungi, because they're kind of ubiquitous all around the world. And we've got them here for sure. Just to show you a characteristic protozoa, Giardia lamblia. This is one that you could acquire in the United States. People who will go camping and get water supplies that are contaminated with these bugs get the gastric symptoms from Giardia. Particularly if you go camping in areas where there's beaver ponds, and the Giardia like to live in the vicinity of beaver ponds. So if you get water around there, watch out for these little guys. They're kind of cool looking. And among protozoa, they have a unique shape. But we, and we also see these in Iowa in well water. So people in Iowa who get their water from a well, if that well gets contaminated with these little guys, then they've got problems too. But aside from those kind of contexts, we really don't see much in the way of protozoa. The classical protozoa was malaria. We used to have malaria in the United States back, I don't know, a couple hundred years ago, but that's been purged. All the mosquitoes that have it are not here anymore. So this one we do see, the yeast. Candida albicans, this one, all humans have this living on them, and we all do. But in small, minute amounts that don't really cause trouble. But if the immune system gets impaired, and one of the classical ways is with prednisone. So a person goes on prednisone, this is where we tend to see it mostly. Candida infections take over in the mouth. It's called oral thrush. You can get it on other mucous membranes too. But it's a budding yeast. You can see little buds there. And I'm only showing one example of each thing. One example of a protozoa, one example of a yeast. One example of a fungus. And the reason I chose this one is because this is something we do see, and I would say from a badness point of view, this is the thing that has caused the most badness that I've seen in my practice. It's a fungus, and it's acquired from soil, and particularly if there's chicken coops and somebody knocks down a chicken coop, and dust comes up, that's a really good way to get histo in your lungs. You breathe in the dust and you got it, and then not only do you have it, but you got it, and if your immune system is good, say it happens to a little kid, it gets walled off and it's just there, and it's kind of living in a dormant fashion, just under check in little lymph nodes in the lungs. And then we've put people on, particularly rheumatoid arthritis patients, on TNF blockers. That'd be like the ones you see on TV, Enbrel, Humira, Remicade, Simponi, Cimzia. Those drugs allow the fungus to break out of its little encasement, its so-called granuloma that the body has it kind of shelled off in, and the granuloma is kind of impaired, and suddenly the bug can take over, and this is bad, and we do see it, and that's why I put this as the chief fungus. But other fungi can do things, but it's particularly in our area. So we, the so-called Ohio River Valley and the Mississippi River Valley, that's where you have histo, and it goes elsewhere too a little bit, but this is the one we worry about. If we lived in Arizona or New Mexico, we'd worry about another one called cocci. That's a so-called desert fever, but that's not relevant to us because that one doesn't live around here. And so worms, I mentioned, we don't see much in the U.S., so I had to pick an example. The only one that really comes up at all clinically is pinworms, and that's because little kids get infected, they have bad hygiene, and these things cause what's called pruritus ani, which is itchy anus. So you can imagine how things happen in a little nursery or a kindergarten class. Kids spread this, and little worms. And we don't see many of the other worms. Like I said, tapeworms and other things are pretty rare. If you go fishing in Minnesota and get a walleye pike with some larvae in the meat and you eat it without cooking it, you might get a tapeworm. But aside from that kind of thing, we don't see worms here. Physical barriers to invaders are important, and the skin, we think of that, skin, that'll keep out the bugs. Well, it certainly does, but it's only 2 square meters of the surface area. Whereas the mucous membranes, that's where the action is, because our digestive tracts, our respiratory tracts, our reproductive tracts, if you add it all together, 400 square meters, a lot more surface area that has to be protected against the bugs. And that's why the GI tract is considered basically the largest lymphoid organ. There's a lot of lymph tissue that resides on the mucosa, which is the boundary between the inside of the bowel and the body itself. So it's very important to wall off those areas with good immune function. So this is kind of the whole talk in a nutshell. And I'm going to talk the first portion about the innate immune system and the second portion about the adaptive immune system. So when I was in medical school, this is all that everybody talked about. It was the whole thing. It was thought that the cure for arthritis was going to be figuring out B cells and T cells, and that was where all the action was, and everybody doing research was doing it here. And I don't even think I heard the name innate immune system. Maybe somebody mentioned it once or twice in some obscure journal, but nobody talked about it. In fact, nobody talked about it really until the last 10 or 12 years, and this has become where all the action is now in terms of research. The innate immune system wasn't really understood much at all. And it's interesting because this is fundamentally probably where it all starts. You know, we were focusing all the research on this. And they're both important. They work kind of together, but it's important to understand the difference. 99% of all animals rely on this only. That's all they have to work with. Vertebrates, like humans, have this one, which has a little extra punch, quite a bit of extra punch. The difference is that this is the initial response. So when you get a little cut on your foot or a splinter in your toe, the innate immune system is right there. It's all over in your skin, and everything. That's so-called dendritic cells and other molecules that are important are right there, ready to go. So that splinter gets in there, and boom, the innate immune system starts working. This takes 1 or 2 weeks to kick in. So even though it's a really good, precise way of killing the bug, the specific bug, you got to wait. So if the bug takes over and a week passes, you're dead. So this is still important. It's really important to be able to at least contain it. It can recognize a wide variety of invaders. Many invaders are kind of have similar molecules on their surfaces, and so these are kind of programmed into the innate immune system to recognize those particular molecules. But it's not every last molecule in the world, whereas this has the potential to recognize every organic molecule that could ever be made under any circumstances in the universe. It's remarkable. So this does the job. It really gets most of the job done, but you've got to have this if you want to survive for any, length of time. This one just handles a small dose of invaders. If you get a big, massive wound that has a lot of contamination, you got problems if all you have to work with is the innate system. This can, if you have enough time, this can completely eliminate the invader. And there's what are called memory cells, which linger and can last a lifetime. So if you see that same bug later, 50 years later, those memory cells will be able to come out and really get the job done much quicker than a week or two. There's also players on this side, which are neutrophils. They're like the foot soldiers. The eosinophils fight parasites. Macrophages are really the coordinating kind of point person in the whole thing. They're kind of like the coach on the football team. And basically, they direct the flow of the whole attack. And they send messages primarily to the T lymphocytes, which are like the quarterbacks on the team. The other thing is, on this side, you've got pretty much the B and the T cells. The B cells make the antibody, a specific antibody for that bug, and the T cells kind of coordinate the effort, or at least they run the plays on the field. But the macrophages are behind the scene, and they're getting the job. They're scouting the opponent, and I'll show you a little bit about what that means. And they're getting the game plan, and then they send into plays. So this is really the essence of the talk right here, this page. So first I'll mention some facts about the innate immune system. This is complement. We didn't know about that long ago when I was in medical school, and it was known that complemented the immune system and its functions and enabled it to do well. Well, what it is it's just a group of proteins that can rapidly annihilate invaders, like really within minutes. And the macrophages are a big Oh, I'm sorry. This is just features of the immune system. I've kind of talked about some of this already. Oh, macrophages are sentinels. They live in the periphery of the skin and of the bowel and all the mucosal membranes. And when they are in the skin, they're called dendritic cells. And actually, on all these areas, they're called dendritic cells. And when I was in medical school, nobody knew what a dendritic cell was. I remember, we'd look at these biopsies of skin, and the professor would say, we don't know what those things do. They're dendritic. Dendrites are like nerve, and it's kind of derived from the term of a nerve fiber, but really it has nothing to do with nerve. Although in a certain sense it does, because the dendritic cells, which are macrophages, and they're sentinels, they're kind of the nerve center. They're the coach of the whole team. Natural killer cells, these are interesting cells that destroy viruses that, well, once a virus gets in a cell and begins to take over that cell machinery, you can't really tell that that cell has anything wrong with it, apart from certain features of the immune system. And the natural killer cells have an ability to recognize those infected cells, and I'll talk about that a little bit later, and kill them. And then I mentioned the eosinophils, they fight the parasites. So complement. This is what I started to talk about. 20 proteins, and they complement the immune system. And it's thought that 700 million years ago this evolved. You can find these proteins, this system, in sea urchins and things that have been around for ancient times. And it's very important because it starts forming in the third trimester of a baby's gestation, and is ready to go at birth. So if a person has a defect, a genetic defect in one of those proteins, they're not going to make it through early life. That's a major problem for that individual. There'll be an infection early on. The most abundant one is C3. We measure this in the lab. We can tell if it's low or high sometimes. And particularly in lupus, it's important because lupus tends to consume complements. So we sometimes see low C3 and C4 levels in lupus. But it cleaves, that means it breaks into two pieces. But when this little guy attaches to an invader cell, it sets up this thing, which I got a picture of, the membrane attack complex. And basically this thing opens a channel into the cell and just pokes a hole in the cell, and that invader is toast once it's got one of those things on it. So it's a very effective way of getting rid of a bad acting invader. The C3b, also this same thing, this same C3b can kind of candy coat an invader. So you got a bacteria and you want to get rid of it, and you just kind of coat it with what looks like candy to a macrophage, and it just wants to eat that thing up. So it's kind of a neat system. Here's the membrane attack complex. So all these, some of these complement proteins come together in a certain configuration, and it's circular. And then on this next slide, it punches a hole in the membrane, and then so all the fluids from around that cell just flow in and just explode the cell, and it's it's devastating for that invader. It's a great way to kill them. So you got this really intense system, this complement system that wants to kill the invaders. But what if it was let loose on the person's own body? That would be really bad. So there's just as many proteins to keep it under check as there are for it to get going. So there's a balance, and you don't want it messing around with host cells. And so, for example, there's this thing called decay accelerating factor, and the human cells have that on the surface. So if there's some C3b that's going to hook onto a human cell, it won't let that happen, and then you won't get this hole punched in the host cell. As an example, back in the day when they were first trying to figure out how to do organ transplant, they did some experiments with other species, and they put, like, say, a pig kidney into a baboon, and what they found was within minutes, and I highlighted that, the transplanted organ was just a bloody pulp, and it was because the complement just went wild on that organ, and it was because the pig version of that inhibitor didn't work in the baboon system. It was just a little different, so it didn't work. But this is just an example of how important that complement is, and it's right there. It's all over in our skin and every other place where a bug might get. So it's important. So continuing on with the innate system, macrophage is the cell I referred to, and it basically phage means eat, macro means a lot, so it eats a lot. And these things basically swallow up bugs, and then the bugs get inside. I've got another slide. So this is the same kind of idea, but it's a little more detailed, where the bug comes in, it gets swallowed up, it goes into a little, what's called a phagosome, and then there's other little membrane enclosed packets of nasty things like hydrogen peroxide and all kinds of things that get dumped into this, and then they start eating away the bug, and the bug then gets annihilated, and then some of its remnants are spit out. These macrophages, these big eaters, are not tidy eaters. They belch up a lot of the stuff, and so when the battle is going on, there's a lot of bug remnants laying around, and that kind of helps the immune system decide what needs to be done by having that happen. There's also other things that messages go to the nucleus, and that sends out what are called cytokines. You may have heard this word because these are really important in rheumatology, because a lot of the drugs that we use block the cytokines. So if you take a blood sample from a person and stain it with standard stain, this is what it looks like. So every day here at Genesis, there's patients who have their blood smeared, looked at, and this is what it looks like with red cells. And here's a white cell, and here's a white cell. They stain blue with this kind of staining. But basically, this has many nuclei. It's called a polymorphonuclear leukocyte, or the other name is neutrophil. And you know, this is what is there helping the macrophage on the front lines. It comes in, and in fact, the macrophage sends out signals saying, hey, I need some help, so that these guys come in and start eating up the bug, particularly if it's a bacteria and a fungus, or a fungus, either one. And then these cells, now this is a lymphocyte. It's just one circular nucleus, looks different than that one. So this one, it could be any lymphocyte. It could be a B cell, it could be a T cell. And we'll just say, for the sake of discussion, this is the natural killer cell. And that's one that seeks out virus-infected cells, host cells. So the eosinophil, this one, the reason it's called that is because eosin is the stain that makes it pink. This is, like Phil means likes, loves, Philadelphia, brotherly love. So eosinophil, it loves this eosinophil, this eosin stain. So it looks pink. All these granules are pink, and they've got enzymes in them that are particularly noxious for parasites. So that's why we have eosinophils, to fight parasites. Now the thing is, they kind of, in the United States, they don't get as much action as they would get in Africa or other places, like I said earlier. So this was the dendritic cell, and we saw these things years ago, and nobody knew what they did, but it's been determined that they're the real big player in this thing in terms of getting the whole thing started. They are great at eating the bacteria, and then what they do after they eat it and chew it out and spit out little pieces, they take some pieces to the adaptive immune system. I'm just going to jump ahead for a moment. The lymph nodes. So what they do is say for example you got a sore throat, there's a bug in your throat, and there's macrophages there that are eating that up and then bringing it. They actually migrate through the lymph channels to the regional nodes, so you feel your lymph nodes swollen. And what's happened is that dendritic cell, that macrophage, is going to the regional lymph nodes, and that's kind of like where all the B cells and T cells come to sample those little fragments of the bug. And it's kind of like a dating bar for antigen to meet up with it's essentially the T cells and B cells. That's what lymph nodes are, and they're very important to get the adaptive immunity going. And you know, a lot of times I've had patients come in and say, "Oh, I got this thing in the lymph node," and the last thing you want to do is take it out. You know, I've had patients ask me, "Oh, can I take out this lymph node, get rid of it?" Well, no, don't get rid of it. It's actually doing what it's supposed to be doing. This is what you want, so you want to let it be. And they are kind of painful if you got a big infection nearby. Sometimes if it's a hand wound, you get a little lymph node right here, or maybe in the axilla. That's why we have lymph nodes, to enable the immune system to get into the adaptive immunity. So just to back up for a minute, so if given time to prepare, the defense can be mounted against any specific invader. And the history is that in the 1790s, this guy in Britain named Edward Jenner came up with this idea that maybe he could enable a person to not get smallpox, which is a really nasty bug, which thankfully we don't have anymore because of the World Health Organization initiative to get it off the face of the earth. But back in the day, it was nasty, and people died from it. Well, it's very similar to cowpox, and it was known that milkmaids used to get it on their hands, cowpox, and then some people thought, hey, if you got that when you're a milkmaid, you'll never get smallpox. So he tested the hypothesis, like, there's a boy, I don't know how he picked this lucky kid, James Phipps was his name, and he was given the milkmaid, there was a milkmaid who had some pus, and it was inoculated into James. And then later, Edward Jenner came to him and with another sample of pus, but this time from a smallpox victim, and he inoculated poor James Phipps, and thankfully the kid did okay. He didn't get smallpox. And so this was the first proof that you could, if given time, you could really gear up the immune system in a really effective way. And basically it was an adaptive immunity that was being used there. So, and the thing about this is, if everything was static and there was no mutations of any kind going on, that'd be fine. But because in nature there's always mutations going on, the bacteria and viruses are able to come up sometimes with new angles on things. And if the animal, the organism, wasn't adapting and changing, eventually the bugs would get the upper hand. So constant adaptation and upgrading. And then about, it's been estimated that 200 million years ago that there was an evolution in fish that led to this huge upgrade. And that has evolved further in humans. And basically it's what I said earlier, this can recognize any molecule in the universe, probably. And basically there's two cells. It's the B cells and the T cells in the adaptive immune system. Every mature B cell makes a clone of antibodies and a single clone. So it's one type of antibody. It's an antibody that recognizes this little characteristic, and that's all that B cell will do, is make them like crazy, and it can just make thousands and bigger numbers than that even. Or it can just recede and just become a memory cell and live a lifetime. So that's it can either gear up and die in a few days after really working hard, or it can just become a memory cell. So this is your standard B cell, B lymphocyte nucleus, and here's how the antibodies stick in. They have these Y shapes, and so basically here's a bug, and here's some sort of pattern on the outside of the bug, and every bug has a different pattern, but this particular bug sticks in right like that, and it can be recognized. And that's a really key thing. And the tighter that bond is, the more affinity that antibody has to the bug, the better the immune response will be. And this is the key thing that really, this is an antibody, and it has certain features, this so-called heavy chain and the light chain, but it's up here where the antigen, that is the bug part, binds to either spot there. And then this side sticks into a cell. And basically there's a lot of different ways that this side can be configured to do different things. Like sometimes you can bond two of them together so that like the two Y's are poking out in this base is stuck together, and that's called IgA. And that's really good for when you've got mucous membranes, because they have a lot of IgA in them. And I have a slide on that later. Let's see, I talked about this stuff already. Oh, so this is antibodies. It's been estimated that you need 100 million different kinds of antibodies to protect from every possible invader. Yet we only have this many genes, so that's like not enough. And so there was a big question for many years, but the guy who figured this out, this guy, I forget his name, it starts with a T, Tekawana or something like that. He got the Nobel Prize because he figured out what happens is you've got the gene. So here's the gene, and you might have for the V, there's different sections. So here would be the gene that would eventually become the antibody protein, this little segment here, and it's got a V3 and a V. Well, there's V3, and it's got a D. It's just got, it pulls one of these, one of these, one of these, one of these, so you can, and then mix and match. So the next antibody might have that one instead of that one. And so you can have these few genes, 25,000, producing 100 million different kinds of antibodies. And here's the different ones I was talking about on that the base of the wall. You got the IgA, that's the mucosal surfaces, that's mucous membranes. IgM is the first one to come out during a initial phase of the immune response, the adaptive response, and then IgG comes out later, and then IgE, that's what defends against these parasites like the worms and whatnot. So this is kind of cool. I'm going to try to make this a little shorter. I will just say that major histocompatibility proteins are really cool, all right? They are elegant and they were unexpected. What they are is billboards, basically. So it's a billboard on the outside of a cell that advertises parts of microbes that are inside the cell, or maybe parts of microbes that come from elsewhere. But the point is, and there's a couple of different classes, but just so I don't get into too much detail, I won't go into a lot of that, but I want to show you a picture of MHC molecules. So it's been said that it looks like a hot dog bun. So you got a bun, this is the bun, and then here's the hot dog. And it basically, that hot dog is the antigen. That's the bacterial or the fungus, the microbe part that's been eaten up and spewed out by the macrophage, and it's being held in place just like that, so that other cells can come by and sample it and figure out if this is particularly something that they could recognize. And if a cell really recognizes this bug part well, then it gets kind of enhanced and it gets an opportunity to proliferate and make daughter cells, and you get a clone of these cells that are really able to recognize their bad acting bug. And this, I'm going to put this in here because co-stimulation is important with one of the drugs we use for rheumatoid. Orencia works to block this. So it's basically signal 1 and signal 2. So you've got the antigen presenting cell with this MHC molecule, that little thing like the billboard that advertises, here's the hot dog, here's the hot dog bun, here's the antigen presenting cell, like a dendritic cell or macrophage, and then here's a T cell, so it comes in and it recognizes. That's not enough. You have to have a second signal, signal 2, and that's these other molecules that come together. And you get both of those to happen, and then you've got this guy gets turned on. He was kind of just lazy and just hanging out, and now he's excited. He says, hey, I want to do something. So now he's activated, and things are going to happen now. It's kind of been likened to a safe deposit box. So you've got a bank safe deposit box, and there's a key that works for all the boxes. That would be like this one. But then there's your specific key that you have for your box, and that's this specific thing. And you got to have both. If you walk in there with just your key, not going to work. If you walk in there with just the teller's key that works for all of them, also it won't work. You got to have both signals to have it happen. And this is a way of kind of keeping the immune system in check so it doesn't go hog wild. So T lymphocytes, there's two kinds. Just briefly, killer T cells do pretty much just that. They find these class 1 billboards that are displaying bugs inside the cell, and then helper Ts, they find these class 2 billboards that recognize bug parts that are outside the cell. And the difference is this will kill the virus-infected cell, and it's by causing them to do suicidal maneuver. There's a thing called apoptosis where a cell is told to commit suicide, and it just kind of dies away. And it's very kind of mild sort of thing, and all the bad stuff inside of it, if it's got virus in there, it gets encased and discarded, the macrophages eat it up and it's gone. As opposed to dying in warfare where you got body parts all over the place, and it's much more nasty. So these guys are very efficient killers by convincing the cells to commit suicide that have viruses. These, on the other hand, they work to secrete a bunch of what are called cytokines. So this is the quarterback, again, of the team. So he's throwing out kind of passes, you could think, but basically he's really throwing out one set of passes. So it's either a group of cytokines that are released that tell the body, hey, look, there's a bacteria here. That's the Th1 response. Or there's a group that there's some worms here that are attacking us, so that's the Th2. And there are these other ones that have been different kind of cytokine signatures that have been described in more recent years. So, last one, last slide. Why mothers kiss their babies. So, in human mothers kiss their babies, barnyard animals lick their babies, but there's something really special about that beyond just the emotional thing, and that is the mom samples the bugs that are on the baby's mouth, the bugs that likely could cause diarrhea for the baby. Now that mom is older, of course, than the baby, so she has had exposures to different bugs. She has memory cells, so those bugs' parts are taken to her lymph nodes, and if there's a memory cell that she has for those, then it'll be allowed to proliferate and really ramp up, and then antibodies specific to those microbes are made, and then they're secreted as IgA antibodies into the breast milk. So the baby now has tailor-made antibodies for that bug that was on them just earlier. So it's just a really elegant example of what's going on with the immune system. So, so basically, again, innate and adaptive immune system, and we got a little time for questions here. So, that's the end of my talk.
Thank you.
Thank you.
Thank you. Does anyone have a question right now?
So what you're saying in part, the immune system can change over time.
Right, the adaptive immune system,
Mm-hmm.
yes, absolutely. And so you can basically get an and the key thing is that antibody. The antibody is really the key thing that allows it to eventually, I mean, there's so many antibodies that can give 100 million antibodies, and there's going to be one of them that has an end here that recognizes that bug, that particular bug part. And if there isn't, they can make more of them too. So that's the adaptive side of it. The adaptive immune system can pretty much do anything if it's operating properly.
Now, certain things, though, is that over the periods of long periods of time for some things, short periods of time for other, or?
Well, yeah, for the adaptive immune system to make antibodies, you gotta have a week or two. And if you don't have 1 or 2 weeks, it's not gonna kick in quick enough. So that's where the innate immune system, which is innate, it's just there, it's ready to go, it's innate to the body. So is the adaptive elements, but the actual way in which the adaptive system comes up with an answer has not yet been made until 1 or 2 weeks have elapsed. Unless there's a memory cell hanging out from a prior exposure to that bug, and then it's good. Then it's much quicker to get ramped up. Yeah.
What part of the immune system breaks down to allow the immune system to start attacking its own cells?
Yeah, well, that's really I didn't cover that at all, and that's certainly autoimmunity. And there's a lot of elements to that, and it's that's basically double this talk. There's just a lot of detail to that question. I mean, it's not one specific part.
A major thing, or?
There's a thing called molecular mimicry where there's a molecule that is in the body naturally, and yet it has a look kind of like a molecule that's in a bug, and there's some confusion on that part. So that's one of the ideas. The immune system gets confused. This molecule looks kind of like a bug molecule. We'll attack it, but in fact it ought not be attacking that because it's a host molecule. That'd be one example. Yeah.
Could it happen by way of a virus infecting a particular organ or group of?
What do you mean exactly?
Okay, just as an example, if you had an infection by a virus or group of that settled in the thyroid or the pancreas or something like that, would that cause like Hashimoto's or diabetes?
No, I mean, once the bug has access to the body, it doesn't really, I mean, there's a concept of a virus that's tropic for the liver. It seems like it just wants to mess up the liver more than other things. But Hashimoto's in particular is just, it's an immune attack. It's the immune system being tweaked earlier, and now it's revved up and it recognizes thyroid, proteins, and starts attacking the thyroid. It's an autoimmune disease. In fact, we can even measure anti-thyroid antibodies in the bloodstream for that particular one.
So would radiation, as an example, maybe cause damage to the thyroid that would trigger an immune response?
Probably, I don't think of radiation really as being the issue there. In fact, I wouldn't think, I'm not a thyroid expert, but I wouldn't think radiation would be the way to do it. In fact, there is a radioactive iodine that is taken up by the thyroid, and the reason it works to knock out the thyroid is that it's radioactive, and it just congregates there and it zaps the thyroid, and then you got a radiation damage to the thyroid. But that's actually a treatment for excess thyroid inflammation. So I don't think radiation is being a culprit there, unless you're talking about you're not talking about thyroid cancer. You're talking about thyroid autoimmune disease.
Right.
Yeah.
Right. Would, if I can ask another, would something like insulin resistance, which I know affects other hormones, would that affect the ability of antibodies to function on that given cell? I mean,
I'm not sure. I know what you're saying.
okay, if you're insulin resistant, you're also thyroid resistant, and you have trouble moving hormones into the cell. Because the outside of the cell is changed by the insulin. That's why it's insulin resistant. It's been changed.
I'm not sure how much that ties in with this. Type 1 diabetes is an immune-mediated sort of a thing, but type 2 diabetes is not really immune-mediated. I'm not a diabetes doctor.
I just
but
wondered if it changed the outside of the cell in such a way that maybe antibodies would also be affected.
Yeah, I don't know the answer to that. I don't think of type 2 diabetes, which is the insulin-resistant diabetes, as being all that tied in with the immune system, unlike type 1 juvenile, which
Right.
is immune-mediated.
Yeah.
Well, any other questions?
Yeah, I know when you talked about the apoptosis,
Yeah,
like where the suicide for the cells.
yeah.
So I was curious, like the killer T cells, basically. So we have biologic medicines that kind of act upon that similar.
Yeah,
what you're talking about.
the idea of a biologic enhancing apoptosis.
Yeah.
Yeah, that is something that researchers have really looked at, because if you can identify bad acting cells that commit suicide, that is a good plan. But,
Okay.
did you have a specific question about it, or just an observation?
Yeah, just, it kind of rang a bell to me.
Yeah, that is a big area. If you've got bad acting cells and you can convince them to commit suicide, that's a great answer. It doesn't leave all that cellular debris, and they just get kind of taken up in a very orderly way by these garbage men, the macrophages. And the macrophages is both the garbage man and the coach too, so
Yeah.
kind of an interesting little role
I
there.
had another question on that, because it came about as far as I was sick here recently. And so the IgM and the IgG, factor when we were testing for viruses. And I guess, can you explain that a little bit?
Yeah, so when we have a person who's sick, and we think it's an infection, a particular one, let's say, we can draw antibody titers, and that's one of the big clues. If an antibody titer comes up and it's IgM, that's a sign that, hey, they've been infected just recently, because it hasn't switched over to IgG. IgG is more of the chronic immune state. IgM is the initial one. It's the first kind of antibody that comes out of the cells when the immune system is revving up. And then later it does what's called a class switch, and it switches to IgG. So basically on these antibodies, it has the same ability to recognize the bug. That doesn't change at all. What changes is down here. Is this going to have a little deal like a clip on to 5 other ones like it, and that would be IgM? Or is it just going to be by itself, and that's IgG? Or is it going to be having a clip down here with another one poking down that way poking out each the other way, that's IgA. And so, you know, the body decides how far along it is in the immune response to send out either IgM or IgG, or if there's something special like the IgA in the breast milk that I was talking about, the body kind of decides. But the bug recognition site is always the same. Once you got a good antibody that can recognize that bug, it's locked in. It's going to be that way for the duration of that plasma cell that makes it, the B cell that makes it.
So is the different viruses, like how long it actually lives?
How long does it last? I don't know the precise number of weeks, but usually by the time several weeks go by, that should start to be fizzling out, and it should be IgG. So if somebody gets a hepatitis vaccine, they've got immunity, and they had the vaccine a few years ago, the series a few years ago, they have IgG in their system. They do not have IgM. It's long gone as IgM. It's IgG. It's just antibodies floating around like that.
Okay.
So, all right. Well, I guess we're done here. Time is up.
Does anyone have any other questions at all?
And then the all-important question we all want to know is what causes our immune systems to go
Yeah.
haywire and attack
Right,
us.
and that actually, my goal for this was just to display the kind of history of the immune system, what it's about. That was the same as your question.
Yeah.
That is a huge topic. There are so many twists and turns on that topic. Just to condense this one into a 40-minute talk is a project, but that is a huge project. So, I mean, maybe that's for another day, certainly.
Yeah.
But yeah, this is how it functions, and you're interested, of course, in how it
malfunctions.
dysfunctions, how it malfunctions, and that's the subject of a lot of research. But I can say that the treatments that we have so far have focused on either blocking the co-stimulation like Orencia does, or affecting these molecules, these cytokines largely, the ones that are kind of being thrown out by the quarterback, the T cell. And that's where most of the action has been. But there's a lot of research being done in the innate immune system, and that probably will be a big part for the future. The immune system, the innate immune system is probably where a lot of the new treatments are going to come from. So, all right, well, thank you.
Thank you.
So you have more meetings left.
Yeah, we do a little bit, so if you're.
All right,
yeah,
well,
thanks
you're welcome.
Thank you very much. I know that's very complicated as far as our immune system and quirky, and that's probably, definitely we can go into detail, and there's not one answer for that, correct?
Yeah, there isn't
I
one
mean,
answer.
there's
a
so
lot
many
of
factors involved.
There's so much to it, and it's hard even to go into much detail except for just to give that example of the molecular immunity.
Right. It makes it
You
complicated.
know, what actually treats it. There's another organ I didn't even mention, which is the thymus, which is probably really important in autoimmune disease because that's an organ that lives in the kind of the base of the neck toward the chest, and it's important in little kids. And as you get exposed to bugs, the cells involved have to course through the thymus, and the thymus is a big black box now in science. It's not really well understood. Stuff goes in and stuff comes out, but the thymus probably has a huge effect on autoimmunity, and that, it's just, it's not known that much about how it actually works. It's a black box.
We're not there yet. Huh?
Pardon?
We're not there yet.
Yeah, we're not there yet. I think we've still got a lot to go, but the amount that we've learned about how it actually works under the best circumstances is very amazing, actually. So,